How does epigenetics influence bladder molecular markers? Imagine your genes as a set of instructions, but epigenetics as the switches and dials that turn these instructions on or off without changing the actual code. In bladder cancer, these epigenetic changes play a big role in shaping the molecular markers we use for diagnosis and monitoring. One of the main mechanisms is DNA methylation, where tiny chemical groups called methyl groups attach to specific parts of the DNA, especially at gene promoter regions. When this happens to tumor suppressor genes like APC or RAS1A, it can silence their activity, removing the brakes that normally prevent uncontrolled cell growth. These abnormal methylation patterns are common in bladder cancer and can be detected in urine samples, making them useful for non-invasive testing. For example, hypermethylation of certain genes can serve as highly sensitive markers to identify bladder cancer early. On the flip side, global DNA hypomethylation, which involves a reduction of methyl groups across the genome, can lead to genomic instability, further fueling cancer progression. Researchers have found that these methylation patterns not only help detect the disease but can also give clues about how aggressive it might be or whether it might recur after treatment. Histone modifications are another layer of epigenetic regulation. Histones are proteins around which DNA is wrapped, and chemical changes to these proteins can influence how tightly or loosely DNA is packed. When histones are modified with methyl groups at specific sites, such as H3K4Me1 or H3K27Me3, they can activate or represses gene expression. These patterns help define different molecular subtypes of bladder cancer, which can have different behaviors and responses to treatment. Understanding these subtypes through histone marks can guide personalized therapy options in the future. MicroRNAs are tiny RNA molecules that regulate gene activity after the DNA has been transcribed into RNA. Epigenetic mechanisms, especially DNA methylation, can turn off or turn on the genes that produce these microRNAs. Conversely, microRNAs can also influence the activity of enzymes that modify DNA and histones, creating a complex feedback loop. When microRNAs are dysregulated, they can promote tumor growth, invasion, and even help cancer cells evade the immune system. Because these microRNAs are influenced by epigenetic changes, they serve as additional molecular markers for bladder cancer diagnosis and prognosis. Clinically, understanding how epigenetics influences molecular markers opens new doors for better detection and personalized treatment. Combining epigenetic markers with genetic mutations like FGFR3 can improve diagnostic accuracy. It also helps identify patients who might not respond well to standard therapies, guiding doctors toward more tailored approaches. While research into drugs that target epigenetic changes is ongoing, these therapies are not yet standard practice. Still, they hold promise for overcoming drug resistance and improving outcomes in bladder cancer. In summary, epigenetics is like the master switchboard controlling gene activity in bladder cells. By modifying DNA methylation, histone marks, and microRNA regulation, it influences the molecular markers used for diagnosis, prognosis, and potentially therapy. This growing understanding is helping scientists and doctors develop more precise ways to detect and treat bladder cancer, ultimately aiming to improve patient care while always respecting the complexity and individuality of each case. Always remember to consult healthcare professionals for personalized advice and treatment options based on the latest scientific evidence.